Hello everyone, I am Suvarna Gaikwad, working as Assistant Professor in Department of Botany of Dada Patil Mahavidyalaya Karjat, District Ahmednagar. So, first of all, I welcome you all in the T.Y.B.Sc Paper-III, that is Genetics and Evolution. So friends, in previous session, we have seen the linkage and crossing over. In today's session, we are going to discuss the new topic, that is construction of linkage map. So, let's start. So, in today's session, we are going to discuss the introduction of linkage map, then its examples with 2-point S-cross, 3-point S-cross and its significance. Okay. So friends, as we know, the genes which are present on the chromosome, so these genes, if they are closely related with each other or closely located with each other, at that time they show the tendency to inherit together, and this phenomena is called as linkage, and such type of genes are the linked genes. But these linked genes does not always remain together. Okay? Occasionally they get separate from each other due to phenomenon of crossing over. So, in this linkage, each gene has a definite order and a particular location in a linkage group. As these linked genes, they form a group called as linkage group. So, in that linkage group, each gene has a definite location and has a particular order. So, linkage is related with the crossing over. Okay, and the percentage or frequency of crossing over closely related to the physical distance between genes. So, if we know all the genes present on the chromosome and also the number of linkage groups of a species, then it becomes possible to determine the distance between two genes, and so that we can determine the gene order also. Okay, and after determining the distance and the gene order, we can construct a map. Okay, so such a diagrammatic representation of distance between linked genes is called as linkage map. So, linkage of gene in a chromosome can be represented in the form of a map, and this map is called as linkage map or genetic map. Okay, so the scientist Alfred Sturtevant was the first who would construct a genetic map. So he used the frequency of crossing over between two genes to prepare a genetic map of X chromosome of Drosophila. Okay. So, for the construction of a genetic map, there are two important factors. We have to know first what is the gene order, and secondly what is the distance between genes. So, if we know the gene order and the distance between two genes, it is possible to construct a gene map. Okay. So, by analyzing recombination frequencies among the progeny of parents, a genetic map can be constructed. So, for constructing genetic map, we should know the recombination frequency. So, the frequency of crossing over, that means the frequency of recombination. Okay. So the frequency of crossing over between two loci is directly related to the physical distance between loci. As the distance between two genes increases, the frequency of recombination also increases. So they are directly proportional to each other. So coming to the unit of measurement of genetic linkage. So the basic unit of measurement for genetic linkage is a map unit. This is also called as centimorgan, which is denoted by cm. Okay. So 1 map unit is equal to 1 centimorgan, and 1 centimorgan is equal to 1% recombination. So for example, if the two genes recombine with a frequency of 3.5%, then they are said to be located 3.5 map units apart. Means whatever the frequency of recombination, that is the distance, that is the map units. Okay. So the formula for calculation of map distance is number of recombinant offsprings divided by total number of offsprings into 100. Okay. Next we will see the gene mapping from 2-point cross. We are going to see the gene mapping from 2-point cross also and 3-point cross also. First, the gene mapping from 2-point cross. So 2-point cross is nothing but a cross which involves two loci. Okay. So they are called as 2-point cross. So easiest way to detect crossover gametes in a dihybrid condition is through test cross progeny. Okay. Let's take the example for the 2-point test cross. So here there are two parents, both are homozygous. So when capital A, capital B, homozygous dominant is crossed with the small a, small b, that is homozygous recessive. So the gametes are capital A, capital B and the small a, small b. As usual, the F1 generation is capital A, small a, capital B, small b. It is the hybrid condition. Again, this F1 generation is test crossed with the recessive parent, that is small a, small b. That is the test cross. So this F1 hybrid produces 4 types of gametes. We know very well that the hybrid test cross, how the gametes are formed. So here 4 types of gametes are capital A, capital B, small a, small b, then capital A, small b and small a, capital B. So these are the general 4 types of gametes, and here the gametes are small a, small b. So after crossing of these both gametes, F2 generation, we got 4 types of offspring, such as these are the combinations as usual. Out of these combinations, if we examine, first is the capital A, capital B, small a, small b, and second is the small a, small b, small a, small b. So these 2 types of combination, these are exactly similar to the parental combination. So these are the parental types. Okay. And another 2 types, that is capital A, small b, small a, small b, and small a, capital B, small a, small b. So these 2 types of combinations are the new combinations which are not present in the parental type. So these are the parental types and these are the recombinant type. In this way, we should know what are the recombinant types. So in the data is given about the number of offspring. As we know, these are the number of parental offspring. So these present in the 160, this present in the 165, this present in the 36 and this present in the 39. So in this way the number of offspring has been given. So from this data, if we have to calculate the recombination frequency, we know the formula for calculating recombination frequency. Recombination frequency is calculated by dividing number of recombinant offspring to the total number of offsprings and multiplied by 100. As we know, there are two types of recombinant offspring. First number is 36 and second is 39. Okay, so they are 75 divided by total number of offspring are 400. So 75 divided by 400 multiplied by 100. So in this way the answer is 18.8. Okay, and the unit is map unit. So the recombination frequency in this 2-point test cross is 18.8, and whatever the recombination frequency is equal to the map distance. So the map distance is 18.8 map unit. That means these genes are approximately 18.8 map unit apart. So in this way we can calculate the recombinant frequency from the 2-point test cross. Okay. Next, this is the gene map. So if now we came to know about the recombination frequency. So if we know the recombination frequency, we know the distance, that is the gene A and the gene B, and the distance between these two genes is the 18.8 centimorgan. Okay, in this way we can locate these two genes and the chromosomes. Next is the gene mapping from the 3-point cross. 3-point cross involves 3 loci. Okay, so here in this figure, here are several different types of crossing over products obtained, which involve single crossover also and double crossover also. As we have seen in previous sessions about the types of crossing over, there is a single crossing over, double crossing over, multiple crossing over also. Okay, so in this figure there is a single crossover also and double crossover also. So these are the parental combinations. 3 genes, capital A, capital B, capital C, all the dominant, and the second type of is small a, small b, small c. So parental combinations, dominant are crossed with the recessive. So here, if the single crossover takes place between these two genes, okay, that is, if the single crossover can be takes place between the genes, that is capital A, capital B and small a, small b. So in that location, at that time the recombinant product is this. Okay, from the crossing over, this is the recombinant product, and with the crossing over in this particular region, there is a swapping. Okay, there is a swapping of these genes. So observe carefully, capital A and small a remains as it is, but the genes, that is capital B and capital C, it gets replaced by small b and small c. So here swapping of these genes takes place. Means that if the single crossover can be takes place between these genes, swapping of genes will be takes place between here. Okay, so the new combinations are capital A, small b, small c, and small a, capital B, capital C. Okay, whereas in the second case, if the single crossover takes place between B and C. That means in this region, at that time swapping will be takes place only in this region, that is as the third gene. So A and B, these are remain as it is, but the swapping of gene takes place between these, that is the capital C is replaced by small c and small c by capital C. That means whenever crossing over takes place in second region, that is between B and C, then the swapping of genes only takes place at the third gene. And in the third case, there is a double crossover. So whenever there is a double crossover occur, at that time there is only swapping of this middle gene. So this middle gene, that is capital B and small b. So capital B here replaced by small b. Okay, in this way A and C they remain as it is, but the B it gets replaced by small b. So here are the locations when the crossing over takes place, single crossing over and the double crossing over takes place. Then what are the different types of recombinations possible? Okay. So now if we have to perform a test cross with the F1, so for the 3-point test cross, the ratio is 1:1:1:1:1, 1:1:1:1:1. Okay. So let's take three genes A, B, C and first make a cross between dominant genes with the recessive genes, and then F1 is test cross to the recessive parent. Okay, that is the test cross. So for that calculation, this is the data given, and from this data we have to determine the gene order and the distance between genes. So if we carefully observe this data, so these are the parental types. Okay, these are the two parental types between three genes. Then these are the single crossover between B and C. Okay, these are the single crossover between C and B. These are the single crossover between A and C, and these are the double crossover products. So if we observe carefully, the offsprings which are more frequent, they are parental type. So they have maximum number of offspring, they are the parental type. Then the offspring which are lowest frequency, that means these 5 and 8, these are the double crossover products, and the total number of offsprings are 1000. So for the calculation of gene order, first determine which genotypes are the parental. Then genotype found most frequently, they are the parental, and from the data it is clear that dominant ABC and the recessive ABC, these are the parental genotypes. Now we have to determine the gene order, and for that we should use the data of the parental genotypes and the crossover products. Then the double crossover gametes are always present with the lowest frequency. So from the table we came to know that these capital A, capital B and small c, and small a, small b and capital C are with the lowest frequency. Okay, but as we have seen, whenever there is a double crossover event, it moves the middle allele from one sister chromatid to other. That means if the parental combination is capital A, capital B, capital C, and if there is a double crossover event occurring this, at that time there is a replacement of middle gene. Okay, so in this way this double crossover event moves middle allele from one sister chromatid to other. So whenever there is a double crossover in these ABC, so this B gene, okay, it gets moved to another sister chromatid. Okay, in this way we came to know that due to the double crossing over, the gene order may be the ACB. We came to the conclusion that there should be C gene which present in the middle, because due to the double crossing over, the B get moved to one sister chromatid ahead. So in this way, this is the gene order ACB. Okay. Now we know that the gene order is ACB. Now we have to know the linkage distance between AC and CB. As we know, the distance is calculated by the formula linkage distance is equal to total number of recombinant alleles to the total number of gametes and multiplied by 100. Okay. So if we have to calculate distance between A and C, at that time we know that, so these are the 81 plus 85, single crossover between A and C, and also we have to calculate the 5 plus 8. So in this way, 81, 85, 5 and 8 divided by 1000 multiplied by 100. So this is 17.9 centimorgan. Okay. Similarly, if we have to calculate distance between C and B, okay, C and B, so these are the numbers, recombinants, 27 plus 30 plus double crossover plus 5 and 8. So in this way, so the distance between C and B is calculated by, we know that it is the 27 plus 30 plus 5 plus 8 divided by total offsprings are 1000 multiplied by 100 is equal to 7 centimorgan. So in this way, the gene order and the distance between two genes can be calculated. Okay. The another example of this 3-point test cross is, suppose that progeny of test cross trihybrid individuals of genotype, that is dominant ABC with the recessive ABC, is as following. So the data is given. Okay. So these are the parental types, dominant ABC and the recessive ABC. Okay. So if we examine, if the crossover takes place between these A and B, at that time swapping will be takes place at the B and C. So capital B, capital C gets replaced by small b, small c. In this way, formation of recombinations takes place. Secondly, if the crossing over takes place between B and C, that is the second case, at that time there is a swapping of only this third gene. Okay, means capital A, capital B will remain as it is, small a, small b remain as it is, but the third gene, that is capital C, is replaced by small c and vice versa. Okay, so that is swapping takes place at third gene. And finally, whenever there is a double crossover, at that time swapping will be of middle gene, that is this B gene. In this case, capital B is replaced by small b. So in this way, from the crossing over, single crossing over and double crossing over, these are the recombinant products. Okay, so here there is the frequency of the parental and the recombinant types. So from this data, if we have to calculate the distance between two genes, first we can calculate the distance between A and B. Okay, For calculating distance between A and B, count all crossovers, okay, that occur in region A. Okay, in region A, the crossovers, that means 18%, and here we can also count all the crossovers for the double crossovers. So in the first region, 18% plus double crossover 2%. In this way, total 20%. Okay, that means 20 map units is the distance between loci A and B. Similarly, if you have to calculate the distance between B and C, at that time count all crossover that occurred in region 2. So we know that in region 2 the crossover are 8% for the single crossover and the double crossovers are 2%. In this way there are 10% crossovers. So the map units are 10 map units is the distance between loci B and C. So finally AC distance is therefore 20 plus 10, that is 30 map units when double crossovers also counted. Okay, and when the double crossovers are not detected, at that time only 26 map units are there from 18 plus 8%, that is the 26%. So in this way the distance between two loci can be calculated. Finally, this is the significance of the linkage map. So linkage map have following uses, such as these maps, they display exact location, arrangement and combinations of genes in a linkage group, okay, which is the most useful for construction of map. Then they are also useful in predicting results of dihybrid and trihybrid crosses. So this is the significance of linkage map construction. So these are the references for this session, and so friends, we will stop here and we will meet in the next session with the new topic. So thank you very much.